Let us now discuss the formation of nucleotide from pentose sugar, nitrogen-containing heterocyclic bases, and phosphate group. There are important characteristics of this combining three molecules into a nucleotide. First one is condensation, with formation of a water molecules. This occurs at two locations. The first one is between the sugar and the base, and the other is between the sugar and the phosphate group. On to the next characteristic. The base is always attached to the carbon 1' position of the sugar. For purine bases, attachment is through nitrogen 9. For pyrimidine bases, nitrogen 1 is involved. The carbon 1' carbon atom of the ribose unit is always in beta configuration, and the bond connecting the sugar and the base is a beta nitrogen glycosidic linkage. The last characteristic is that the phosphate group is attached to the sugar at the carbon 5' position through a phosphate ester linkage. There are four possible RNA nucleotides differing in the base present. We can have adenosine, cytosine, guanine, or uracil. And four possible DNA nucleotides differing in the base present. We can have adenosine, cytosine, guanine, or thymidine. Let us now discuss nucleotide nomenclature. All of the five names end in 5' monophosphate, which signifies the presence of a phosphate group attached to the 5' carbon of ribose or deoxyribose. Preceding the monophosphate ending is the name of the base present in a modified form. The suffix "ocene" is used with purine bases and the suffix "idine" with pyrimidine bases. The prefix deoxy at the start of the name signifies that the sugar present is deoxyribose. When no prefix is present, the sugar is ribose. Again, the naming starts with the prefix deoxy if the sugar present is deoxyribose. But for RNA, there would be no prefix present. This will be followed by the bases whose suffix will be replaced. Again, for purine bases, we will be using the suffix "ocene", and for the pyrimidine bases, we will be using the suffix "idine". For instance, the purine base adenine will be now named adenosine, and the pyrimidine base cytosine will now be named as cytidine. The 5' monophosphate will complete the nucleotide name. The nucleotide abbreviation comes from the one-letter symbols for the bases, the use of MP for monophosphate, and a lowercase d at the start of the abbreviation whenever deoxyribose is the sugar. Nucleic acids are polymers in which the repeating units, the monomers, are nucleotides. The nucleotide units within a nucleic acid molecule are linked to each other through sugar-phosphate bonds. Primary nucleic acid structure is the sequence in which nucleotides are linked together in a nucleic acid. Because the sugar-phosphate backbone of a given nucleic acid does not vary, the primary structure of the nucleic acid depends only on the sequence of bases present. The specific backbone structure for deoxyribonucleic acid or DNA is an alternating sequence of phosphate and deoxyribose, while for RNA it is an alternating sequence of phosphate and ribose. Take note that the nucleotides are all oriented in the same direction. Do take note also that the bases are attached to the first carbon of the pentose sugar, or the C1 prime. This order is important to determine the genetic information of the molecule. The alternating sugar phosphate chain in a nucleus or nucleic acid structure rather is often called the nucleic acid backbone. After the addition of the base, the nucleotide is now formed. Just as the order of amino acid side chains determines the primary structure of a protein, the order of the nucleotide bases determines the primary structure of a nucleic acid. As mentioned earlier, a nucleotide chain has directionality. One end of the nucleotide chain, the 5' end, normally carries a free phosphate group attached to the 5' carbon atom. The other end of the nucleotide chain, the 3' end, normally has a free hydroxyl group attached to the 3' carbon atom. By convention, the sequence of bases of a nucleic acid strand is read from the 5' end to the 3' end. So with this example, thymine is the first base from the 5' end, followed by adenine, guanine, cytosine, adenine, and another cytosine on the 3' end.